All right, for the 59,000 take, JK, just kidding. Anyways, it is the introduction to antibody structures and immunoglobulin structure, as well as their function in terms of their expression. So let's get into first the overview, and then we'll actually get into the overview of the actual somatic recombination during the actual B cell development, and then we'll go a step further and talk about how the changes that occur after the actual antigen encounter can improve their differentiation and their variability via somatic hypermutation, isotype switching, as well as the affinity and maturation. And then we'll finish off with the specialized functions of each individual IG. So, starting out with a quick overview of the structure, let's talk about the difference between the heavy chain and the light chain. We're going to see the yellow chains are going to be the light chains, and then you're going to see the heavy chains being the green ones. So why that's important is because they're basically composed of different types of amino acid sequences. Heavy and light chain obviously varying in the amount of amino acids that make up the actual sequence itself and the actual sequence order. So we're going to get into, just like a normal amino acid, you have a C terminus and an N terminus. At the bottom there in the green, you see C termini, then you see the N termini. You also have, in terms of the inner, that's where the actual heavy chains are, going up, up, and down. And then getting down here using the same picture but a different color scheme. That's is when we're going to basically explain the most important thing, which is going to be these guys, these actual antigen binding sites. Now, this is incredibly important because you're going to see that these antigen binding sites, as we know in most relationships with human bodies, structure determines function, and so is the same for this. So we're going to look into a variable region as well as a constant region. This variable region is up here in the top part, which uses both light and heavy chains. Light one's out here, heavy chain right there, heavy, light. But then the other second half of those light and heavy chains, as well as down here for these heavy chains, is what we call the constant region. This is incredibly important because the constant region itself is actually the effector function, or the effector region, if you want to call it that. Lastly, we have what we call the hinge region, which is where the disulfide bonds are, right in here, same thing up there, and that's important because you'll see that we have a protease function that comes in as an enzyme. It's going to basically snip. As we go to the next slide, you'll see that, and why that is important is because it actually snips at the disulfide bonds, cuts them, and it breaks apart and makes these into 3 fragments, 2 of them being the same, meaning that they're the fragment of antigen binding, and then you have this one fragment, which is the fragment of the constant region, as we just spoke about. So, as we get into this, you will see that what that does is almost like keys is the best way to use it as an analogy to explain it. So, if you can see it, you can look at this key going into this antigen binding site right here from the bacterium, and it's more like a rectangular square. Then we have a triangle, and then up here we're going to have a circle. So it's kind of like this, that we have keys, so they're the same family of item, but they unlock different things or are used for different types of locks, but they all unlock something, right? So, that being said, these different structures are allowed to function differently of what type of antigen they're able to bond to and neutralize, right? Now, they all have a variable region, which we can see. The variable regions are up here, as we talked about, up there, and that's incredibly important because that gives it the ability to have what we call variability, or hypervariance at times, which gives it a greater repertoire to respond to different types of antigens, but also being very specific still. Now, when we look into this, just really quickly, you just see that you have heavy and light chains that actually contribute to these 3 hypervariable regions, and there's 3 hypervariable regions actually inside of one variable region itself. And it's made up of heavy and light chains, and this creates what we call the Fab arm, which is what you see right there. So going into it, just to give you a little better of a depiction of the 3D structure, this could be confusing to some, but basically you're seeing what we would call your effector function down here, which is going to be your constant region, and then more of your variable region up top. Alright, so here's a little bit more of a precisely organized diagram of it, or labeled per se, and this is basically going to give you, basically showing you how the IGs are folded upon itself, like in the shape itself, and they're basically very compact, and they are a stable domain of amino acids. So you can see this and how stable it is. So we have constant light chain, variable light chain, constant heavy chain, constant heavy chain, constant heavy chain with a variable heavy chain over there. And then same thing, this is before the actual protease, because you can still see these disulfide bonds, right? So going a step further, that hinge gives it the variability to be folded, right? It can actually bend and move. So as we go a step further, we're basically going to talk about the antigen binding site. So right here, this is exactly what we're talking about with the antigen binding site. It can vary in shape, and if it varies in these shapes that you see there, all maybe similar related but still distinctively different enough, that's going to change the composition and the structure as this figure basically shows in 410. And then we know again, structure determines function, so it gives us some specific versatility and allows us to have different isotypes, which we're going to be discussed here a little bit later in this video. So basically, as the last part of the intro, we need to make sure that we know we need diversity, and we need diversity to cater to different types of antigens, and this comes from what we call random recombination, and this is basically the rearrangement of a gene, which is also called, it's going to be also called the somatic recombination, which we're about to discuss here in just one moment. And basically, this is going to use things like RAG gene and recombination enzymes. Again, we're about to really get into it. And this is the whole process of recombination, is to make these functional genes. So here we go. This is the next section here, and we're getting into how this actually happens and how you end up getting to the somatic recombination. So on the left side, you see light chains. Just real quick, you're going to have three regions: the V, the D, and the J. V for variable, D for diverse, J for joining. You'll see that just the light chains over here, boom, boom, you only have the V and the J, and given the structure, again, it's much larger for variable because it does have more variability. Then you're only going to have one round of somatic recombination in order to get to the VJ join rearranged DNA down there. Over here on the heavy chain, however, you have two rounds of that, and picture it like having a party. You can't have a party without DJ, so you have D and J recombined first right here through somatic recombination. Then you're going to go a step further, and you're going to have somatic recombination one more time, and then D says, hey, I want to have a party, and you have a DJ, so let's have a party. So the V and the D and the J, they have a big antigen binding party, and what they're doing is basically making these genes that are going to allow them to code for specific binding sites, basically, and to have that variability that they need specifically. So going a step further, this helps definitely with the isotype switching and whatnot later on, but right now, early on in this B cell development, that's why it's incredibly important. And we're going to talk about the RAG genes right here. Okay, and real quick with the RAG genes, to keep it basically simple, we basically have things that will correlate this whole recombination process, and there are the recombination activation genes. So RAG1, RAG2, we'll see that up there and up there, and you will also see 12 base pairs and 23 base pairs. Why is that important? Because the recombination sequence has to happen in between, based on that 12 to 23 base pairs in between. If it doesn't, it's not going to happen. So how does that happen? Well, you're also using what we call a VDJ recombinase enzyme, which is almost like a cut and paste enzyme, and that's going to give the ability to really add that diversity, because the antigen binding site of the immunoglobulin itself will basically have germ-like DNA like we just talked about there, and have the ability to cut, paste, and rearrange when exactly necessary. And then once it actually creates the successful desired gene, then we'll have the ability for it to be a functional gene, and then it'll say that it is in the necessary, not conformation, but the final structure per se, that's going to allow it to have that desired variability, which is exactly what we're trying to do, because it's more challenging before you've been introduced to an actual antigen or pathogen per se. So what happens then at that point, this is the point when it just becomes binded on the B cell receptor surface, as you can see depicted here in this diagram. Then we go a step further, and we're going to talk now about the overtypes of the changes that happen when you finally get an antigen encountered, and you get into somatic hypermutation, and you have isotype switching, and you also have affinity maturation. So what happens here is you see mutation frequency. So it goes up, 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 up right here, and it's basically gonna target these rearranged gene segments that encode the variable region. That's what gives it the variability. So it happens here, it targets this variability region, boom, goes all the way back, affects transcription. Now that's going to affect the actual reproduction of these gene rearrangements. Now we go a step further and we look at a couple things: the structure, the enzymes, and the RAG1 and the RAG2. So the enzyme is the BDJ recombinase that we just talked about, and then we'll also see what happens when we use specific enzymes with the BDJ recombinase, like we just said. And then you also have this alternative mRNA splicing, which makes IgM and IgD. Each B cell, though, equals basically an immunoglobulin of a single antigen that's specific to it and is made as a membrane-bound form of the B cell surface, which we just showed going up right to there. So here is just a basic example, just to talk a little more specifically about what it looks like when someone actually gets immunized, and the difference between early on and then 2 weeks after your primary immunization, of what it looks like to see how many different receptor regions you have in the heavy chain and the light chain, and how you have IgM and IgG, and if you just see how many different of these cell receptors you have, is quite a lot more, increasing the actual variability, which is a very favorable thing for immunity, so you can have more immunity to a wide range of potential pathogens or harmful antigens. Now, when we look at this and see the mutations, right, it can be generally thought of as a negative thing, but it can be also a positive thing, because these mutations allow us to have the variability, which is again the whole end game, especially after now that we've been exposed to an actual pathogen. So, going a step further here, you will see that you basically have different C regions, or constant regions per se, and that's what gives us the ability to have a little more variability, because the constant region, if you recall, down here and over there, back up there, that gives us the ability, because that's the effector function, and when those are changing, or when they are different, they have different effector functions, and that's very useful, because different effector functions will be different functional variable structures that will give us the capability to respond to multiple antigens, which is a lot better for us, so that each one has their own specific niche. It's like having a team and having the right pieces of the puzzle, that everyone has their job and role, but together, collectively, they're a very strong team. Now we look at this and we're going to see pentameric IgM and then monomeric IgM. Well, IgM, pentameric versus monomeric, you'll see the 5 different for pent all the way around it, around that joining chain, and that's important because that's going to improve its overall affinity. So that's the affinity maturation that we're talking about. So going a step further now, this is when we get into the actual, you have the different isotype switching taking place, and then you have the, even for IgG, for instance, the 4 subtypes that's incredibly important within this, that are different, but they can also be complementary to the function of the antibody binding structure, and then therefore it increases the overall B cell diversity, which is huge, which is exactly what we want after the antigen encounter. So the next time we encounter this antigen, it doesn't have the ability to bring about the same inflammatory response, or bring about, the ability to really wreak havoc on the body itself. So as we go just a step further in here, we're just going to go back in, and you will see that basically with IgG specifically, it has IgG1, IgG2, IgG3, IgG4. Now, to remember all these different subclasses and the different isotype switching that we have from immunoglobulin, you can remember something called damage. IgD for D, IgA for IgA, and then you're going to have, for the M, it's IgM, and then you're going to have IgA, so you still remember that, and then IgG, IgE, and then just IgD, but we've already talked about that. So now we're going to talk about just individually what they do. So IgD, to start, the class of immunoglobulins that have the delta heavy chains, and it appears as on the surface of Igs on mature naive B cells, but its function is actually unknown, other than the transcription of IgD is coordinated with that of IgM. So we also know that the sensitization of basophils occurs with IgD, and you can see that right up on here boom, boom, boom with plus, plus, plus. So this is a good chart to see the difference of each one of them. Now we get to IgA. This is the class of Igs that have alpha heavy chains, which is great, and you see that up there because this happens to affect neutralization, which is incredibly important to neutralize the pathogens or the antigens that are in the body. It's a dimeric IgA antibodies that are the main antibodies present in mucosal secretions, and they have a monomeric IgA that's actually present in the blood itself. It also has a little bit of function of opsonization. You can see just a 1+, as well as the activation of the complement system, and then down here obviously the transport across the epithelium when it's in its dimeric form and not just monomeric, as opposed to down there with the diffusion of extravascular sites. Now we get to IgM. This class of Igs, they have the M heavy chains, which is related to the lowercase of the mu for the Greek alphabet, and is the first immunoglobulin to actually appear on the surface of the B cells, and the first antibody secreted during the immune response. So in that kind of innate immune response, if you want to talk about it like that, and it's secreted in its actual pentameric form, which is what we had seen just prior up here into the pentameric form. So it also helps huge with complement activation, as we'll see right through there with the pluses on it there, and right there, complement system. And then we'll go a step further into IgG, which has a bunch of these subclasses. And real quick, we'll just talk about how they have the gamma heavy chains. And IgG is the most abundant class of Igs in the plasma. It does a lot of stuff, as you can see. We have neutralization taking place. We have opsonization taking place. We're going to have activation of complement systems. We're going to go a step further and have diffusion into extravascular sites. Each one of the IgGs have that, which is huge for us to be able to get into the external sites. And then we have neutralization all across the board with IgGs, which is incredibly great because that's the first line of defense, basically. Then IgE is the class of Igs having the actual epsilon heavy chains, and IgE is involved in reactions against internal parasites, particularly helminth worms, and in allergic reactions. And then in combination with that, it has sensitization of mast cells, and that's what we get into a little bit later in chapter 8 and 9. And you talk about how basically IgE is able to help coat these different parasites, and then that gets combined with the actual FcER1s, and you're able to have degranulation occurring of the mast cells, which just produces a whole cytokine storm and basically allows you to neutralize the cells that you need to that are actually hurting you in terms of the antigens, and basically activates a whole cascade of inflammatory cycle and immune response necessary to get rid of the actual antigens that are needed. It is also the least abundant in the serum, and it does also help affect the sensitization of the actual basophils, and then it does, in combination with that, it does also help affect the sensitization of these mast cells, which is exactly what we're talking about for degranulation. It was a lot of information, and I hope you got something out of this. That's it.